thought it was coming here. So students, today I will be explaining about linkage. Before linkage, certain additional points we should know. One is, who was the one who provided experimental proof for the chromosomal theory of inheritance? In 1902, a theory was propounded by Sutton and Bovary, and the theory was the chromosomal theory of inheritance. And the proof for the chromosomal theory of inheritance was provided in 1933 by a scientist, Thomas Hunt Morgan. Okay, this scientist, what he did is he experimentally proved this theory called chromosomal theory of inheritance. So therefore he becomes father of experimental genetics. Now for all his experiments, he has used fruit fly. So it is an experimental animal for Morgan's experiment, Thomas Morgan's experiment. So this fruit fly is also called Drosophila melanogaster. This is the scientific name of this fly. This fruit fly depends on, usually why it is named as fruit fly, it is found around the rotten fruits and all, it will be more. Okay, so that is where it is commonly found. Now, now these are the following reasons why did he choose the fruit fly for his experiments. Not first reason why he has chosen for his experiments is simple synthetic medium is required for the growth of the fruit fly. That means the nutrition for fruit fly can be a very simple synthetic medium which is very cost effective. Got? And sometimes, even though if there is no simple synthetic medium, the fruit fly can survive with a rotten banana or rotten apple. That is how it can survive in a simple food. Therefore, that was also one of the reasons why he has chosen the Drosophila. The second reason why he has chosen the Drosophila is it shows several hereditary variations. It means that Drosophila have different mutants inside it. Okay, that means it shows different mutants. That means different characters are there, so that he can study those characters for inheritance to see how those characters pass from one generation to next generation. So that's what was studied. Okay, so that is the reason. Variety of insects are there. That means variations are there. Because of those, he can study according to his convenience the various mutants. Then second, single cross gives rise to many eggs or offspring. So that is also a reason why he has chosen this. Single cross, that means single mating, leads to many offsprings. Or you can say gives rise to many offsprings. So that means this is also one of the reasons why he has chosen it. Then it means that a single mating can produce many eggs. Then that egg will hatch and give rise to many insect offsprings. And therefore, he can study those offsprings for how the character is getting transmitted from parents to offsprings. Then it has a short lifespan. It has short lifespan of about 2 weeks. So when the lifespan of the insect is too short, he can study several generations within a short period of time. So therefore, that is also one of the reasons why he has chosen it. Then there is a clear differentiation between male and female. That means there exists sexual dimorphism. Male can be easily identified and female can also be easily identified. When identification is easy, then every generation, how the trait will pass in the male and how the trait will pass in the female can also be studied. So therefore, this is also one of the advantage of using Drosophila. Then sixth reason is it is already scientists studied Drosophila in the past, so its genetics was well known. So since already a known fruit fly whose genetics is well known, it will be very easy to study. So as a result, it is an added advantage for the scientists to do experimentation. So this is the also another last reason which can be given for this fruit fly. That is, its genetics was well known. But so these are the list of reasons why Drosophila melanogaster was used in the experiments of Thomas Hunt Morgan. So again in brief, in 1902, chromosomal theory of inheritance was propounded, but still there was no evidence for this theory. And therefore, experimental evidences was given by Thomas Hunt Morgan in 1933. Next, I will teach a topic recombination, which is also called crossing over. What is recombination and what are the steps involved in recombination? These topics will be covered. So let's take recombination. To understand the concept of recombination, you should know what is homologous chromosomes. So this is consider this is the one of the chromosomes. Then every chromosome will have another pair which are similar. So therefore there is another chromosome which is very similar to it. And these are called as pair of chromosome. Identical pair of chromosomes they are called. Okay. The name for these chromosomes is homologous pair or homologous chromosomes. Homologous chromosomes are identical to each other. They are similar to each other. That means this chromosome is very much similar to this chromosome. And in these two, one will be arising from the father, it's called as a paternal chromosome. Another from the one is called as from the female, that is the maternal chromosome. So therefore here, these two pairs, identical pairs, are called as homologous chromosome. Such type of pairs will be there in human beings. So human beings totally have 23 pairs of chromosome. Okay. In that, 22 will be autosome and the 2 will be allosomes. So the homologous chromosomes undergo a phenomenon called recombination. So how this recombination will occur? In order to understand recombination, one of the chromosome would be tainted here or to be colored here to differentiate. Now you can see that the maternal chromosome is being taking the shaded color. The paternal chromosome is not shaded. So by this you can differentiate which is maternal chromosome and which is paternal chromosome. Here, you should know that each chromosome has two strands. This is one strand, this is another strand. Similarly, the maternal chromosome also has two strands. Now, these chromosomal strands are called chromatids. Totally four chromatids. 1, 2, 3, and 4. Four chromatids are supposed to be present. Okay. The first chromatid, this one, two chromatids which are belonging to the same chromosome. How it is identified is it is bound to a common centromere. So this chromatid is called sister chromatids. These two chromatids which are bound by a common centromere is called as sister chromatids. And in case of maternal chromosome, even these are called as sister chromatids. But on the other hand, if you compare a chromatid, for example, if I assign it as A and then I will compare with this B. So these two do not belong to the same chromosome. So these two will be non-sister chromatids. Even these two will be non-sister chromatids. So if I assign letter A, B, C, D, in that case, A and C will be non-sister, A and D will be non-sister, B and C will be non-sister, C and D also will be non-sister chromatids. It means that simply what you should know is the chromatids which do not belong to the same chromosomes are called as non-sister chromatids. Okay, that means chromatids of paternal as well as maternal chromosomes when compared are called as when recombination occurs between them, then it's called as non-sister chromatids. Now, what will happen in recombination is, before how the recombination occurs, you all should know when do actually recombination occurs. Recombination occurs in meiosis. And you know that meiosis has two main subdivisions, meiosis 1 and meiosis 2. So recombination occurs in meiosis 1. Okay. And within meiosis 1, there are further steps like prophase 1, prophase 2, metaphase 1, metaphase 2, like that. The recombination is going to occur in prophase 1. And also, within the prophase, there are several steps: leptotin, zygotin, pachitin, diplotin, and diakinesis. So recombination will occur in the pachitin stage of prophase 1 of meiosis 1. This is where actually recombination occurs. So what happens here is, before recombination, the two chromosomes pairing should occur. That means the two chromosomes should be paired. Okay. So what should be there is, first the two chromosomes should be paired, and then a segment of chromosome should be broken, and another segment of the chromosome also should be cut, and then they should be exchanged. That means segment of chromosome, one of the chromosomes should attach to the other chromosome, and segment of this chromosome should attach to the other. So what happens is there is an exchange of chromosomal segment. This exchange of chromosomal segment itself is called as recombination or crossing over. So therefore here, these are the non-sister. Now, in between whom the recombination occurs is always between non-sister chromatids. That's why I have highlighted as to which is the non-sister chromatid. So therefore, exchange of chromosomal segment occurs between the non-sister chromatids of the homologous chromosome is the definition of recombination. So exchange of chromosomal segment chromosomal segments between non-sister chromatids of the homologous chromosome. So exchange of chromosomal segments between the non-sister chromatids of the homologous chromosome or homologous pair, you can say. So this will be the definition of a crossing over, which is also called recombination. Now, what you came to know, definition of recombination you came to know, and then you know where and why exactly recombination occurs. And one of the primary significance of recombination is, due to recombination, there is variations among the offspring. You know that recombination occurs in meiosis, and meiosis is the one that is responsible for production of sperm and ovum. Okay, so they are like connecting between the two generations. During what happens is recombination will bring about variations in gene combinations. As a result, the offspring will show variations, and variations are raw material in evolution. So now, what happens is first a pairing should occur. The two chromosomes actually will be far apart in the nucleus. Pairing should occur. After pairing, there is crossing over occurring between the strands, non-sister chromatids. What happens here is the strands are going to cross over before exchange. So suddenly they won't exchange, they are going to cross over, then there's a cutting, exchange and rejoining. So here what happens is crossing over occurs. So as you can see, when crossing over occurs, X shape is created. So this crossover point is called chiasma or chiasmata. So now what are the steps involved in recombination? So recombination involves three steps. One step you can tell cutting. Second, you can tell exchange. And the third is rejoining. And the third is rejoining. So this you can obviously know. If these two chromosomes has to exchange, there should be first pairing. Okay. Usually after pairing, there is a crossing over occurring between the two chromosomes, creating X-shaped structure. Okay. After crossing over, what happens? First, the chromosomal segments will be cut. At which level they are going to be cut is homologous sites. Cutting occurs at homologous sites. What are homologous sites? The identical sites. Suppose cut is made here, even cut also should be made at same level. So I can tell molecular. Okay. So at same position the cut is made, and this identical position is called as, or identical site is called as homologous site. So that is about the first step in recombination. Okay. Then second is exchange. So what happens once this segment is cut, this will be detached from rest of the chromosome. And even when this segment is cut, this is going to be detached from rest of the chromosome. Next will be exchanging. The segment which is being cut is going to be exchanged with the next chromosome, and this is going to be exchanged with the other. So what happens? Segment of one non-sister chromatid is exchanged with the segment of another non-sister chromatid. Thus, these are called recombined chromosomes because they have segment of another chromosome, so they are called as recombined chromosomes. So that itself is the process involved in recombination. Okay. So what will be means what is exchange is mere segment, and you should know that once the exchanges occur, the segment can detach from the chromosome. It can detach from the chromosome. So to prevent it from detachment, it should be permanently attached to the chromosome. For that, what is necessary is rejoining is necessary. So cutting, exchange, and rejoining. These are the three steps in recombination. And these three steps are performed with the help of an enzyme. And that enzyme is named as recombinase. So recombinase is a biocatalyst called as it is an enzyme, multi-enzyme complex that does the work of cutting, exchanging as well as rejoining of the chromosomal segment and carries out the process of crossing over or recombination. Now after this, these chromosomes will get separated. So after the recombination, the chromosomes are going to get separated. Now what happens? Suppose the recombination is over and these are resulting the already recombined. I can tell you how to. Now what has happened? This is a shaded one again. It will take a portion from the other chromosome. A segment is got exchanged now. And here a segment of other chromosome is exchanged. So what you can see is exchanges occur at this point. Similarly, exchange can occur at many points. So only one exchange will not be seen. So exchange means that is the region where the exchanges occur. So likewise, many chiasmatas can be formed. Multiple chiasmata can be formed here between the two non-sister chromatids. So that is but for convenience sake, I have only shown the single exchange. But likewise, similarly like this, many exchange will occur between the non-sister chromatids. So this was about what is called the recombination. Now the same chromosome is going to enter recombine. Now these two chromosomes, this one and this one, they are recombined now. Right? And these chromosome itself will enter into gametes. Now if I show the gametes, say 4 gametes are formed from meiosis, for example. These are the 4 gametes. And each chromosome will enter into one gamete. So now you can see this is a recombined, it consists of recombined chromosomes. And in case of this also, it consists of a recombined chromosome. But this is again not recombined, it is more parented. So these two gametes which are formed will be recombined. It means that paternal and maternal characters or genes will be mixed. And therefore, if the offspring is born with this recombined chromosome containing, of course, recombination will be very extensive. So therefore, sperms will have many recombinations. It means paternal character will be mixed with maternal character. As a result, resulting in variations. It is also one of the reasons why variations will occur in nature. And as I already told, variations are raw material for evolution. So next is linkage. For that you should know a phenomenon called linked genes. Linked genes. Now what is this linked genes? What is the phenomena involved in this? So linked genes are those genes. Suppose first I draw a chromosome. Right now, since I am not showing recombination, I have drew a single chromosome, single stranded in a single stranded form. Here, there is one gene. Let's say it is X, and there is another gene called Y. Now, these two genes are present in the same chromosome. Okay. Such type of genes which are present in the same single chromosome, they are called as linked genes. So, what is the definition of linked genes? Genes located on the same chromosome are called as linked genes. Genes located on the same chromosome are called linked genes. They're called as what? Linked genes. And the phenomena is called as what? Linkage. So that is how, what do you mean by linked genes? Linked genes gives us an idea of inheritance. What is that? When linked genes are there, the chances of crossing over will be less. So therefore offspring resemble more like parents. So parental combinations will be more, whereas recombinant variations will be very less, or recombinant offsprings will be less in percentage. So that is what is explained in this linked genes. The NCERT gives a definition, different definition for linked genes, and different in sense, meaning is the same. It is a physical association of genes on the same chromosome. Is also called as linked genes. Now, in this, there can be two types: the complete linkage as well as incomplete linkage. Incomplete linkage, you can also tell partial linkage. Now, if the chromosome, if the two genes or more, not necessarily the gene should be only two. There can be many genes on the same chromosome. So, all that, those genes will come under a single group. Okay. If the two linked genes are very close to each other, very closely placed to each other, usually such that there is absolutely no recombination, or there is no recombination, we can tell. So such type of linkage is called as complete linkage. So in complete linkage, what happens? Parental characters directly go to offspring, and offspring will be more like parental characters. So that's called as linkage. That's complete linkage. But many a times, the linked genes are present in same chromosome, but they are far away from each other. In the same chromosome only, they are far away from each other. When they are very far away from each other, same chromosome, there is chances of exchange of chromosomal segments or recombination. That means this X can go to the other homologous pair, the other one can come here. So there can be exchange of homologous there can be homologous chromosome, and therefore recombination can separate these genes. It can separate this linked genes. When they are far away, when there is a chances of recombination, therefore we say it is incomplete linkage. So it can also be called incomplete linkage. Okay. Then certain points relevant to linkage. If I now show, this is one chromosome, which is one of the strand of the chromosome that is undergoing recombination. For example, it has gene A and B. The other homologous pair, it has recessive form of the same gene. Okay. Now suppose exchange will occur between these two strands belonging to homologous pair. Now if there is a cut in this position, this cut is called identical site, called as homologous site. Now exchange will occur means this segment is going to get exchanged here and this segment is going to go to the other side. So as a result, what happens is down the side, down to the other chromosome, whereas the dominant one goes into the other chromosome. What does this do? What does it prove is the recombination can separate the linked genes. Okay. So next is more the linkage, more is the, that means more the distance. Now next point I will tell you about distance between the two genes. What does this explain? Distance between the two genes. Understood? If two genes, okay, are far away from each other, then recombination percentage also will be more. And if the distance between the two genes is very less, then chances of recombination will be also less. That means you can arrive at a general conclusion. What is that general conclusion is? The distance between the two genes will be directly related to percentage of recombinants. This is another point which you should know. See, when the linked genes are very close, as such they pass at least for more than 2 generations, they keep on passing from one generation to another generation without recombining if the distance is very less. But if distance is far, still there is a chances of recombination and therefore parental characters may not pass from one generation to next generation. So that is nothing but what is called the relationship between the distance between the two genes, that is linked genes here. With distance, it should be distance between two linked genes. Distance between two linked genes is directly related to percent recombination. So this is also another point which is very important. Now Morgan performed the experiment using Drosophila. What he has done is he has taken two of the fruit fly. One of the fruit fly, it is supposed to be the female fruit fly, and then he has crossed it with the male fruit fly. So the female fruit fly is female fruit fly is crossed with. Now what are the characters present in the characters present in female fruit fly is that The body of the fruit fly was yellow in color, and the eye of this female fruit fly was white. And then what happens is the male fruit fly, it is supposed to have the brown body instead of yellow body. So body color was brown instead of yellow body. Whereas the eye color was red. These are normal features. So these were the two fruit flies that were crossed in one of its experience. One of its experiments. Now this was crossed, and this resulted in F1 generation offsprings. Then F2 generation led to F1 generation led to F2 generation. Now in F2 generation, there were two groups in the result of the F2 generation. Some of the offsprings resemble parental characters. Okay. That means parental characters, as you can see it here itself, parental characters means character of parent. That is fruit fly which have yellow body and white eye, brown body and red eye. Those are called parental characters. But some were there, some of the offsprings were non-parental. Some of the offsprings were having non-parental characters. Non-parental characters which are not exactly like parent. It might be yellow body color but red eye, or brown body color or white eye. So these are called non-parental characters. Now non-parental characters can be called as recombinants. Non-parental characters are called as recombinants. Sometimes it is also referred as recombination frequency. Okay, frequency of recombination, which is usually expressed in percentage. Okay. Now in this, 98% in F2 generation, 98.7% of them were resembling the parental characters. They were having which characters? Parental characters. And remaining 1.3% were non-recombinant. It means that they never resemble the parental character, but cross resemblance. So that means yellow body, red eye, brown body, and white eye. These are called recombinants, non-parental or recombinants. Now, clearly in the F2 generation, what you are seeing is it is not giving Mendelian ratio. So it is not giving rise to which ratio? Mendelian ratio. Okay. It is a dihybrid cross. So why it is called a hybrid cross? Because it consists of two characters. One is Y, Y plus. Another one is W, W plus. These are the. This is 1, 2, 3, 4. At a time, they are considering two characters means it is a dihybrid cross. So when it is a dihybrid cross, it should show which ratio? It should have shown 9:3:3:1 ratio, but it is not showing it. Instead, which characters are more here? Parental characters are more, whereas recombinant characters are very less here. So this itself indicates that the two genes are linked. So it is a outcome of a linked genes. So therefore, we should also know linked genes give rise to more parental types, whereas less recombinant types. Now, when absolutely no recombinant types is there, the linkage is called complete linkage. Other ways it is called as incomplete linkage or partial linkage. So, therefore, this was about the linkage concept of linkage and some of the important points which you should know in the concept of linkage. Okay. Okay. The second experiment was again, I should not have rubbed it slowly. So one of the fruit fly, okay, one of the fruit fly was having miniature wing, and miniature the wing size. It means let me tell it very gently. In the second experiment, he has taken the wing size of one of the female is crossed with male. So apart from wing size, again it is eye color. Eye color. So first will be wing size is miniature wing size. Another will be red color. This is actually white color eye. And whereas the male will be normal phenotype. Male will be the wild type. Now this again, as I already told, results in F1 generation, and further it is going to give rise to F2 generation. Again, same thing. Parental combinations will be very high, whereas recombinant percentage will be very low. Recombinant percentage you can tell, or recombination frequency you can tell, that will be very low. This again indicates linkage. So how much percentage was it? 62.8% was parental type, whereas 37.2% is recombinant type. So that is what is the experiment carried out by Morgan. So Morgan did these two experiments. As I already told, distance between the linked genes is directly proportional to the percentage of recombination. So therefore, one more scientist was there. His name was Alfred Sturtevant. He utilized the data obtained through experiments and then prepared a chromosome map. What he does in chromosome map is he is able to find out the sequence of the gene on the chromosomes. He is also able to find out what is the distance between the two genes in that sequence. So therefore, how is that done? So Alfred Sturtevant was the one who prepared chromosome mapping. Now I shall give a simple example. In this chromosome map, the recombination frequency is taken in order to find out the distance between the two genes. So the distance between the two genes can be calculated, is measured through a unit called centimorgan. Centimorgan it is called. This is the unit to find out the distance between the two genes. centimorgan it is called. And this centimorgan is obtained from the percentage of recombinants that are experimentally performed. Okay, in an experimentally performed in an experiment in the F2 generation, based upon what is the percentage recombination, based on that distance between the two genes can be calculated, and the unit is centimorgan. So, giving a simple example as to how the chromosome mapping is done. So, if I say gene between A and B has say 20% recombinance, and genes between B and C is say 5%, and gene between A and C, the recombination that is recombinance of the between the gene A and C is if it is 15%, then chromosome mapping can be done. Now, if I look into the percentage of recombination, you will come to the sequence. So first what is called gene will be A, and then between A and C is the next gene. Here, C can be here. So you have to do rough work. So between A and C, the percentage recombination is 15%. So what you have to do is, it is nothing but 15% is nothing but say 15 centimorgan distance between A and C. Now C also continues with B. So distance between C and B will be 5%. So distance between C and B, for example, this will be 5%. So 5% is nothing but 5 centimorgan. Then coming to AB will be the total distance. So summation, that means additive it is. That means 15 centimorgan plus 5 centimorgan together will give rise to 20 centimorgan. So what does it indicate is distance between gene A and C is 15 centimorgan, between C and B is 5 centimorgan, and between A and B is a total 20 centimorgan. And very much simple is that it is additive. So this is the distance. Now what is the sequence? First gene, for example, is A, the second will be the C, and the third will be the B. So arrangement of the gene is ACB. This is the arrangement of the gene ACB. The B is not at the center, C is at the center. So AB, ACB. This is the sequence of the gene, and also what we come to know is distance between the two genes. This is how chromosome mapping is done. So this completes an example that shows how the chromosome mapping is done. And the first person to do the chromosome map is Alfred Sturtevant, and he was the first person to prepare the chromosome map in Drosophila.